So thanks everyone for joining us for our live Lipid Maps webinar today, which is going to be presented by our guest speaker, Michael Witting, entitled The C. elegans Lipidome, What We Know and Don't So Far. My name is Valerie O'Donnell. I'm based in Cardiff University in the UK. Today's webinar is being sponsored by Avanti Research. We're grateful for their ongoing support. And now I'm going to introduce Michael. So Dr. Michael Witting studies applied chemistry, taking functional direction into biochemistry at the Georg Simon Ohm University of Applied Sciences, Nuremberg, Germany. He obtained his PhD in analytical chemistry from the Technical University of Munich and had his habilitation from the same university. And since 2021, he heads the metabolomics team at the Metabolomics and Proteomics Core Facility at the Helmholtz Center, Munich. His research focuses on the development of novel analytical approaches for LC-MS-based metabolomics and lipidomics and tools for improved identification of metabolites and lipids. And for 5 years, up to 2025, he was on the board of directors of the Metabolomics Society. Michael has a big interest in C. elegans as a model organism for lipid metabolism, as you can tell from the title of his talk today. And that's actually great from our point of view on lipid maps as well, because it gives us great opportunities for curating in new lipids that get discovered in that organism. And I highlight a recent Nature Communications paper with Campo, Piano, Bode and Dierking in 2025, in which they showed that sphingolipids, I guess not from C. elegans, but from Pseudomonas infecting C. elegans, protect from bacillus infection, which sounds like a really interesting story. And I'm sure that resulted in numerous new structures, especially since you're working with Dominic, who has got lots of new bacterial sphingolipids that he keeps curating in as well. So I'm looking forward to hearing the talk. And I think what Michael said he's going to cover is what makes the C. elegans lipidome so different. What lipid classes or categories that are not found in mammals are present in C. elegans, and how can new lipids be identified, I guess, using mass spectrometry, particularly focusing today on C. elegans. So thank you very much for coming today, Michael, and I'll hand over to you now for your talk.
Thanks, Val. Thanks for the invitation. I'm really looking forward to this. And these are two topics very close to my heart, C. elegans and lipidomics. So these days, I'm also doing lipid analysis in humans and other organisms, but C. elegans was always close to my heart ever since my PhD, because it's a real fun organism to work with. And as Valerie said, the title is What We Know and We Don't Know So Far. And there's already the big fat spoiler coming up. So if you would scale this roughly, it probably looks like this. So this tiny little bit that we know already, but there are many, many things we don't know so far. So the talk today is a bit of an historic view from how did I get into these things, to some newer applications until some newest applications on lipid identification. So you might have heard bits and pieces out of this, and at the end there will be some kind of a conclusion to say, okay, what do we need to move forward actually with this C. elegans lipidome? Brief agenda, what we're going to cover, very, very brief introduction, something I like as analytical chemist, reproducibility. That's something I'm very keen on. So it was also for C. elegans a question I was always asking myself, how reproducible is the entire thing we're doing in here? The curation of some C. elegans lipids, this is something we also did in collaboration with EpiLipidNet and a few others, and it's a still ongoing process. It's a never-ending story in here. Identification of novel C. elegans lipids that we did and tools we used for this. And then there's a question what we still don't know yet. And very, very brief conclusion. And I'm happy to take questions towards the end if there are any. Very, very brief introduction. And I titled this Worms Fat and LC-MS-MS. So that's basically my life since my PhD. I've been moving to C. elegans during my PhD, where we used it as a model organism for infection of Pseudomonas aeruginosa, which turned out to work much better than our cell culture models. So ever since then, I stick to C. elegans as my favorite hobby organism to work on. For all the people not familiar with C. elegans, so it's a soil-dwelling hermaphroditic worm. It's a nematode. You can find it in all moderate climates. So even here on rotten fruits, if you have a compost in your garden, like I have downstairs, here you can find probably some C. elegans where it's feeding on the bacteria in the compost in there. It's microscopic in size, so an adult worm is about 1 millimeter long, 65 microns in size. So if you have very good eyes, you can see adult worms crawling on the agar plates. And for all the other things, all the other larva stages and the larvae stages, you need a microscope for this. It's hermaphroditic of nature, and depending on the temperature of cultivation, there's a specific portion of males in the population, but the most part is hermaphroditic, which makes it very nice for any kind of genetic studies you're planning. It has about 2 to 3 weeks of lifespan, so also very good for aging studies in here. And it has a boom and bust lifestyle. So actually, an adult worm can lay up to 300 eggs, which within a very short developmental cycle actually develop into further adults. So even if you need a lot of material, biomass, for your analysis, C. elegans is very easy to grow in large amounts. The most important point why it's used as model organism is that a lot of human disease genes and pathways are present in C. elegans. They are present maybe in a simpler copy, a simpler version of it, but they're present. So C. elegans is actually a model system for development, neurobiology, host microbe interactions, aging, Alzheimer, nutrition, diabetes, and you name it. Actually, many, many more applications in there, especially development, since the entire cell lineage of C. elegans is known. But covering the topic today, one thing I realized very quickly is that it has a real rich lipidome, but only partially characterized. And this is some kind of a lifetime work I want to do, and currently also working on to map out the lipidome of C. elegans in here. So this is where we started off. Actually, it was published in 2014, but we worked way before with that, where we had our first method optimized for lipidomics. So it's always lipid profiling, reverse phase in here. And you can see actually here, we started with very low numbers nowadays. We would say, okay, 2012, 1,250 features at max detected, only a few hundreds with MS, but it was reproducible, and this method we published is still our workhorse. We use it for the analysis. But at that time, we had very limited knowledge about the C. elegans lipidome. Which classes are present? What's the composition of the lipidome? Only a few things have been published. And then there was the question, are there any weird lipids? Knowing already that from some publications that it might not entirely reflect something we would expect working with mammalian lipidomes, for example. And this is where one of my big interests comes from: lipid identification, better tools, faster tools for lipid identification for working with the data. And with that, I actually want to jump into a topic we did a few years after. It's about the reproducibility of these things. So as usual with omics studies, there are papers that are actually having the same results, some are contradicting each other. So the question really for us was, okay, how reproducible is this entire thing? And for me, as Val already mentioned, is I'm a chemist. I'm an analytical chemist. I had a detour into biochemistry. So I know a bit of both worlds. And for me, metabolomics and lipidomics is actually perfect. It's between analytical chemistry and the biochemistry in here. But mostly I'm a chemist, and when somebody talks to me about a model system, for me it's a very defined input and you get a very defined output. But then reading the protocols, how to cultivate C. elegans, it basically says, okay, prepare this buffer, this media, grow E. coli overnight, spread it there, and let the worms feed on it. Where I said, okay, that doesn't sound very reproducible for a chemist, right? And even you have things like here, bactopeptone here from DIFCO or bacto-agar, where we already know there's a huge batch-to-batch difference in there. And this all dates back to the original protocol from Sidney Brenner. So this is actually from his paper on the maintenance on C. elegans. So, and there's a chemically defined medium for C. elegans at the moment, which really allows us to do in-depth biochemistry work. There have been a few described, but none is really working in here. So the question I was asking is, how is this actually influencing the metabolome and the lipidome? So to see actually how big the differences are, I asked 4 labs I was working with at that time if they could grow me a bunch of worms. And with a bunch of worms, I actually mean some replicates of the wild type N2 and some replicates of a mutant, the DAF2 mutant, which is one of the best characterized mutants we have in C. elegans. And the idea was basically do we get the same biomarkers from all of these laboratories? And if not, what's the reason in here? Or can we recapitulate the same biology from these kind of comparisons? So we even had one lab that was so kind to grow 2 batches to see actually within one laboratory how comparable are these things. So there's a very elaborate protocol about QC samples, a total QC sample, and so on. I don't want to go into details too much and directly jump into the results. So when I talked to the biologists in here, they said, yeah, you know, DAF2 is such a huge mutation, changes a lot of things. DAF2 is actually a fat phenotype. It has enhanced lipid droplets and a higher lipid droplet count, so meaning more triacylglycerols stored in there. They were believing if you do a principal component analysis, we have wild type on one side, we have the mutant on the other side, and the lab might be on the second thing. Whereas I'm not sure about that, right? So let's have a look. And turns out that I was right. Actually, what we see are distinct clusters of the individual laboratories in here. So you can see actually lab A is clustering together, and if you color it, this is always the self-fulfilling prophecy of the PCA, if you know the groups. We can see slight separation of DAF2 and N2 in here. We see lab B up here, and very interestingly, we see lab C, D forming a cluster down here. And lab C and D were actually from the same city. They are both based in Lyon, and they are only separated by one street. And they were actually kind of sharing the chemicals. So they have the same vendors, so they have the same bactopeptone, and even they were sharing their E. coli. So they said, hey, look, mine looks weird. Can I have yours? Yes, of course, back and forth. So they were actually very similar. This is something we can also see in the lipid profile in here. But I was actually interested in getting the biomarkers of these things, comparison with DAF2. Are they up or down regulated? So one thing we did actually was looking into the fold change of things, and they should go always in the same direction. So what we plotted is actually the fold change between the DAF2 and the N2. So you can see actually everything that's blue is downregulated in DAF2. Everything that's red is upregulated in DAF2. And we were comparing the fold changes of one lab against the other. And here you can see, for example, lab A batch 1 and 2, you have the best correlation of these fold changes, whereas for some others, the correlation is a bit weaker. But what's very important for me is actually to see that there is no lipid that actually suddenly changed the quadrants. So it means downregulated in one laboratory and upregulated in the other laboratory. So this actually shows that the general trends we can recapitulate. It might be not the exact same fold change, it might be not always the exact markers, but there are specific lipids and trends we can actually get across all these different laboratories. And one example is here, where we have actually the sum of the triacylglycerols as a very, very rough measure for lipid droplets. We actually know that DAF2 is a fat phenotype, so they should always have higher levels. And you can see this here in red. It's for all the labs the case, except for lab A1. But we know already that these worms were close to the starvation, so they were already using internal reserves. Here the levels were not as high as the others. And also generally for lab A, they are not as high as the others, or the differences are not as pronounced. But we see that. And another marker that has been described already is actually PC4010, which is downregulated in DAF2, and we actually found this in all the different laboratories. So that basically told us, yes, it is reproducible to a certain point. It's not like you get fold change 2 here and fold change 2 there, but you can get the same markers, saying even though if we have a very rough protocol, as I said, for a chemist, where they use bactopeptone, things are reproducible. And with that, actually, we moved into many, many more studies, and these studies are currently under re-investigation, as I said, for mapping the metabolome. So we have been working with great collaboration partners doing lipid analysis on very different studies, and this actually helps us to annotate now in depth the lipidome, what we're currently doing in here, because we have here a huge diversity from different genetic backgrounds, from different studies, from different laboratories in here. And that actually brings me to the second part in here, or the third part, is about the curation of C. elegans lipids. Of course, lipid maps has been very, very helpful with the things, and certain lipids specific to C. elegans made it up into lipid maps these days, and they're also linked to C. elegans in here. But when we started off this entire endeavor, we had no bloody idea what to expect in C. elegans. So what we actually did is we leveraged what is called a genome scale metabolic model. And we used this genome scale metabolic model to predict lipid species based on the biochemical knowledge or on the pathway knowledge that we have in C. elegans. And we're currently refining this tool to be actually of use for different other organisms, so we can use it in a reproducible fashion. So how does it work actually? So if you have a look into such a genome scale metabolic model, typically reactions are encoded like this. So you say you have a fatty acid CoA, and you take here a lyso PA, and this yields actually a CoA and a PA. But these reactions are on a very generic level, right? So this fatty acid CoA could be anything. This lyso PA could be anything, and well, CoA is fixed, but then you get any kind of PA. So we started actually to write a code that enumerates all the possibilities, giving some fatty acid input and with the CoA input to generate all the different versions in that. The advantage of that is, of course, you can always do combinatorial synthesis in the computer of your lipids, but with that we also have the link to potentially producing enzymes in here. And furthermore, the code is working in such a way that it also simplifies some lipids. So we can either report this network on a full structural level, or we can actually break it down to something like species level, where we have a more generic version of these things. And this gives us the possibility to predict lipids, and we can directly map out which enzymes might be related to this. And to give you some ideas, so this is from a nice review from Watts and Risto. This is on C. elegans lipid metabolism. So we have here very classical. We start from glycerol 3-phosphate to lysophosphatidic acid to phosphatidic acid to deacylglycerols, and they could be either then be used in the phospholipid biosynthesis, either here via CDP, here for PEPC, or we go for the PAs with CDP-DAC pathway for PIs or cardiolipins, or they go into triacylglycerols. To give you an idea, I only took this very short stretch where you say, okay, that's only a few reactions. But if we take this into our network approach, you would see that if we use 16 different fatty acids as starting points, we get over 4,500, so close to 6,000 metabolites, lipids, and we have about the same amount of reactions. So this is actually at the moment for all genome scale metabolic models a computational overkill, but it gives us a good list of lipids we can expect based on known biology that we have so far. And you can see also there are certain things like here, C. elegans has these methyl transferases in here. Where also there have been nice work from Dominik Schwutke on this, showing that there are also the monomethylated versions, so monomethyl PE, monomethyl dimethyl PE present in here. So we have with that some way of handling this, but this is just a prediction. So the question was, how can we actually compare this? So we took a very naive approach. So we scaled everything down to lipid species at the moment. We curated actually the literature, and I recently started to recurate a few of the papers and looked into all the figures, supplementary information, and mined out all the different lipids that have been in there. It was also part of an AP-LipidNet hackathon we organized here in Munich. And the question is how to compare this. So we boiled everything down to species level, because we said, okay, that might be for starting point the fairest comparison. And I generated plots like these, where each tile actually shows the presence of a specific lipid species with a specific number of carbon and a specific number of double bonds. So if you see this blue tile there, this means, okay, at minimum one lipid species with this specific composition, number of carbon, number of double bonds has been detected. And I used in parallel a lot of my own measurements we used on the Bruker-Maxis, on the timsTOF Pro 2, and upcoming now also on the new Bruker Tims Metabo as well. So we have 10 data sets. We have over 750 LC-MS runs we could use. And we're going now for in-depth annotation in here to also compare this against our own measurements, where we can look back also into the raw data and really check, is this identification true or just a bogus identification? Something we can do only for a few selected papers where the raw data actually has been shared. And to get you an idea, actually, this is now for PCs, PS, and PIs, the same plots. So we have on the top level here the prediction. And what you see actually for PCs, PS, PIs, the predicted lipid species looks exactly the same, because what we're not doing at the moment in the prediction tool is take into consideration different fatty acid pools for the different lipid classes. This is something we have to work on, but this is something we have to refine based on common knowledge that's in the literature so far. The second line, you see actually the complete literature. So this meaning if a lipid has been detected only once, it's in there. So there might be one hit wonders, only detected under very specific conditions, only detected by one specific laboratory. So we filtered the things also down to say, okay, look, everything that has been detected between 5 and 9 papers, or detected in 10 or more papers. And then also here, these are some measurements from one data set on the timsTOF Pro 2, actually where we have the own identifications. And what's striking me is actually that, for example, for the PCs, we see that actually prediction and literature in our own measurements are matching quite well. So there are a few things that have been predicted we didn't measure. There have been also a few things that haven't been predicted but have been measured. So we need to refine in both ways, but overall for PCs it looks quite fitting. But if you go to PS and PIs, you actually see that especially if we filter down to having it detected in more publications, we're horribly over predicting. So it looks like not all of these species we're predicting are present, or at least not at levels that they are detectable with our standard LC-MS approaches. So this is something where we need to refine in here and get a workaround to really have also this information somehow as feedback into the prediction, some kind of a scoring algorithm or so. But this is something we're actively working. And by the way, I'm doing this with Thomas Nake from the PTB in Berlin together to get this done. But one thing we can't do with that at the moment is novel lipids. And this is going to be now the biggest stretch within the talk. How can we identify novel lipids in C. elegans? So what we used actually for these things, since they're all run on Bruker mass specs, in the meantime, we also have data sets from other mass specs as well, is we used Metaboscape and the rule-based lipid annotations, and we validated this rule-based lipid annotation in here. And from version to version, it got much better. But this only gives you the lipids where we know the fragmentation rules and they could be implemented into Metaboscape. But the question is now, what about novel lipids? What is in there, and what can we do? What can we find? And here, and I want to show you 3 selected examples that we found and where we're currently working on. One thing that was very interesting when we looked into one of the data sets is actually we found some very unusual Fachwas, so fatty acid esters of hydroxylated fatty acids in here. And you know that they all have been linked, for example, to diabetes in here. And they typically have lengths of 16 or 18 carbons for both of the fatty acids in here. We used here actually Lipid BLAST, which is an in silico generated library, where I said, okay, let's don't waste any time. Let's use this and then have an idea. And we found actually C9 containing Fachwas. And this was where I said, okay, that is weird on the first glance. I checked the MS spectra. They were actually fitting. So you see, actually, this is the C9 hydroxylated fatty acid. This is the, for example, C14 in here. So the MS spectra were actually fitting. Overall, also the retention trends were fitting very, very well in here, that you see this increase with the increase in chain lengths. But the question was, what's going on in here? So we have a chromatogram here. We have here again the trend line that we see, and also here the ratio of actually the easterbound acyl over the hydroxyacyl, and this is actually indicating this ratio. If you look into the literature, at different positions of the hydroxyl groups, and so there are things like we can have an omega -1, or anything in between in here. But what caught my attention is actually when I was drawing such structures is that this looked somehow familiar in here. So these omega -1 hydroxy fatty acids are found, for example, in these different signaling molecules, which are called the ascarosides, and they are very, very active. And ascar-10 actually here with the C9 fatty acid is the most prominent signaling molecule and the most abundant one in C. elegans. So this tells me that is actually a bit more than chance of finding these things. So there might be some real biological validity behind this. So we're currently trying to identify these in much more data sets to see are they detected only under specific conditions. I have to admit they are actually, although they have very nice chromatogram, very low in intensity. So we need to find ways of enriching them in here. So this was one thing. So this was in the database. These were things that have been known, but there were also other examples. And one example that caught my attention is actually this very nice paper here from Boland and al. I really like. And actually they were looking for sterols. So they were searching, or they were using actually sterol deprivation in C. elegans over multiple generations, and were looking for signaling molecules, a molecule that help C. elegans coping with the sterol deprivation. They were always saying, okay, there might be something sterol related in there. What they found was actually phosphorylated glycosphingolipids. So I asked myself, these very specific lipids, are they only produced under this very specific condition they used in this single paper, or are they generally present in C. elegans? So what we did is we went into the literature, we checked these things. Some of them have been deposited lipid maps in the meantime, so this is a bit outdated. In here, it's a very unusual class because if you have a look, C. elegans, and this is for one of the key points to take home for you, C. elegans, for example, produces a very weird sphingosine base, which is a C17 isobranched sphingosine base. And I remember talking to Michael Witting when he was asking that, Michael, we don't detect any sphingolipids in C. elegans. I don't see them. I was asking what they used, and they used MRM transitions, which are based on C18, and I said, okay, just remove one carbon and they will be there. And he found them afterwards in here. And the other thing is that we have this normal looking glucosylceramide, but they have this decoration in here attached to the sugar. You have either phosphatidylethanolamine or a monomethyl phosphatidylethanolamine residue. There have been no reference spectra available, so not electronically deposited. We looked into the SI. You can find the fragmentation patterns of these in the supplementary information of Boland et al. So what we used is actually we took this information and transferred this into a mass spectrometric query language, which was used to search in, for example, 2 different data sets now for peaks that show this fragmentation pattern. To explain you what the fragmentation pattern looks like, so you have this lipid, it actually loses the head group, so to say, the glucosyl part with this decoration, and it produces 2 different fragments in here. For this is not a monomethylated one, so we have some kind of a motif we could look for. But just because these peaks are actually present doesn't mean they are belonging to the same lipid class. Could be random features all over the place that by chance have masses within a specific error range. So what we leveraged here is actually retention time, and since we worked on an ion mobility instrument, also collisional cross-section. So we had one data set we generated on a Bruchromaxis, which we used for mining this, and the other one coming from the timsTOF Pro 2. And we checked actually the retention time. So no standards are available for these things. So you have to rely on all the information. And actually, many of these features showed this fragmentation pattern. Some of them were actually too low to yield good MSMS. But combining this now into these trend lines actually helped a lot in identifying these new lipid classes in here. And the same is actually true if you go for the collisional cross-section in here. So we can see we have the retention time differentiation, and also we have here the trend line along the collisional cross-section. And one important point I would like to raise here is actually that you see this point. So there are the monomethyl and the methyl, the non-methylated and the monomethyl versions. Of course, they can differ in the location of the methyl group. It could be either in this head group or it could be in the side chain leading to the same mass. And what you see actually is that CCS-wise, they are actually very, very close in the collisional cross-section. So for me, it's very important and a good example to point out that actually chromatography in combination with eye mobility is the thing that you would like to use for lipid analysis. I get asked quite often, and Michael, eye mobility is now so good these days. Can we just drop this bloody chromatography and rely entirely on eye mobility? Where I said, no, there are two reasons. First of all, for real close structural isomers, we might not have the eye mobility resolution, which is actually shown here. I could only differentiate them because they were eluting at different retention times based on the CCS value. And the other thing is we still have ion suppression in here. So I'm a big fan of chromatography to separate these things out. We took this actually even further. I didn't have it in here, and we were looking for the precursors in here. So this is actually a phytoceramide or a phytosphingosine in here, where we see we have this second hydroxyl group inside, something that only has been partially described in C. elegans. So we could also, using MassQL, find the precursors. We could find the hexasyl versions and also the normal ceramides in here, which are either the precursors or degradation products of these. And we were looking in the meantime also in further data sets. And one of the big data sets we're currently analyzing is the developmental stage series, where we were going through the different larval stages to young adults. And you actually see that these lipids are not only present in all the stages, it looks like they have regulation over the series. So from L1 to L2, they're going down, and then they're going gradually up again until the young adults. So this is something we are interested in. We're looking for lipids that are following similar trends. So it looks like they also have biological importance, not only analytical importance or curiosity in here. But watch this space. So we're hoping, we're currently annotating all the lipids from these data sets. It's a real in-depth analysis where we have a few thousand features detected, and we're now going one by one to see what they are. The question that's still open is here, this will come later again in the summary slides, is how is actually this lipid here produced, this phytoceramide. So there is one enzyme which is a candidate for this, the gene is TTM5, which is actually doing this step here of the desaturation in here. But there's also an intermediate step with the hydroxylases, and I believe that this might be the missing piece we have, but not entirely sure at the moment. You see there's also another homologue which might be responsible for this. So there are many more things we need to investigate. And one other example about novel lipids I would like to cover now is actually what we called chypinea glycerophospho N-acyl ethanolamides. We found them actually in one data set when we were looking for lyso-PE. So we saw actually multiple peaks in here. But if you look into these a bit closer and check the MS-MS fragmentation, so we have the one here, which doesn't show a very classical PE fragmentation. So this is negative mode. What you would expect is to see the fatty acyl chain in there. So this showed a completely different fragmentation pattern. For example, these two, if you're familiar with that, is a phosphate group. And you would see that these two are actually belonging to glycerol phosphate in here. Whereas if you go to the other two main peaks, you would see that it's 253, which is 16.1, as you all probably know, working with lipids. So these are actually the classical lyso-PEs that we would expect also to be present in C. elegans to a certain extent. So we're wondering what these lipids are, and we found a few publications, and they have been described, for example, by Aryutkin and the group of Frank Schroeder here, also doing some network analysis. So we went further down the road and checked them, and they're isomeric to LPEs, and they are potential intermediates on the route to N-acyl ethanolamides, and they have been described in C. elegans actually. These are potent signaling molecules, not only in C. elegans, but There are C. elegans. For example, they have been found in the starvation response. And if you look into this, what we call the chipineas, these are the ones here that have been described down here. So what you have is from PEs, you isolate the head group and you end up with a NAPE, an N-acyl phosphatidyl etanolamine in here. And you can either directly hydrolyze them to cleave off the etanolamine residue with the acyl chain, or you go via this side products. Interestingly, there has been some in vitro activity tested. There are two enzymes, NAPE1 and NAPE2, which have this NAPE phosphatidyl phospholipase D activity. So this route might be known already, but we detected these things. So to give you the structures, the idea, we have an SN1, SN2 LPEs. We have the chipineas, which are having here the side chain on the head group, so to say. And there are also hexosyl variants of these where you have an additional hexose moiety in here. Fragmentation-wise, this is something that we just described already. Is we have the phosphate group, we have here the glycerophospho, and we have some other fragments. Also, the glyco version fragment a bit different, so you have an overlap also with some fragments from actually the sugar in here. So one of my PhD students, Lisa Salzer, developed an annotation tool, which we call APEX in here, which combines multiple networks to find these lipids. And one of these things is using mass difference networks, which solely based on MS1 features on top. So we did some molecular networking using GNPS algorithms, for example. We could find based on the MS-MS, but also on the MS1 alone, we could find a few candidates in here. But again, since they are not annotated by an MS2, the question is really, are these real things or are these just artifacts? And this leads me to the next part. Again, the use of retention time. So we could use retention time here for these things on top. Typically, retention time is used at a very late stage of metabolite and lipid identification, very typically when you want to match against the reference standard. But this homologous series actually holds a lot of valuable information, and people in the environmental research, for example, have found this, and this is from the PFAS community in here, and thanks to Nico Kessler from Bruker, he made me aware of this paper, where they actually use the homologs and the consistent retention time behavior to boost their identification rates. And this is something that is also done in lipidomics for checking the retention time trends. But the question is, how are you going to do that if you don't know anything about the lipids, right? You have no idea about the fragmentation pattern, or you might have an annotation pattern idea, fragmentation pattern idea, and you only find a few candidates and you want to find more of these things. So we actually measured chipineas on the new TIMS Metabo on an alpha version where we had access to it. So there have been many enhancements in this one, and it's called TIMS Metabo, but it also delivers for lipid analysis. This is something I can tell here. And the most important piece for me was actually here the software to really leverage this information. So again, we went into this data, we generated, and we used now RT trend lines within TIMS, within Metaboscape. And this has been leveraged for the rule-based annotation because it's first checked on the MS2, and then it's checked if there are consistent retention time trends along the things. So if you have a PC which is not following actually the rules of the PCs, it's flagged and it's potentially removed. But again, here we don't have any rules. So the question was, how can we do that? So we established the fragmentation. We could find the fragmentation in here. So this is again example of a chipinea 14. This is a hex chipinea 15.1. So we have the usual suspects that we were actually expecting to be present in here in this fragmentation pattern. But what now the people from Bruker actually implemented in here, and this is shown in this small video, is a homologous series search, which you can use interactively. So for example, for the paper with this ceramides, I was doing this entire thing manually within an Excel, checking do they follow the same trends, plotting this with Excel and R, going back and forth between different software tools. This is now possible actually here within Metaboscape by selecting a few candidates you have, and it automatically draws you, as you can see, this trend line. And you can use this trend line actually to search for other potential candidates. Are there any candidates lying on this trend line? So you can have some hypotheses. You can draw even weird trend lines. We had ones for the elongation, we had for the desaturation, so you can find actually cross points of certain things. And this is, for example, now for the hexacyl variants. And it calculates you automatically the position where it would expect the next member of this to be present. And if you have even associated sum formula, it calculates you the sum formula difference between the things. And by this, you can actually very interactively work with the data and get some idea if there are any potential candidates that might be fitting to the thing. So you can see here we have some examples which are actually very close on the line, but we also have some isomers which might be outliers. But you see here, for example, a predicted one and one that was actually fitting very, very well within these things. And if we look into the fragmentation pattern, we could actually see, yes, this was also fitting. Now the video starts all over again. I just want to point out, this is actually very helpful. As I said, I did this in Excel and R before, but it's a real interactive tool which allows you to interactively mine your lipid and metabolomics data to get some better ideas of what exactly is going on. And this is really helping for the identification of novel C. elegans lipids. And I hope this gave you some ideas now what you could do with C. elegans. And I believe there's still tons of lipids where we have no bloody idea what they are, what their functions are, and if they are structurally related to any things that we know. But that's another question, what we still don't know. One thing we realized is actually in C. elegans, we have no idea how specific the enzymes are. Just coming back to this short stretch that I showed you for the prediction in here, we used all the fatty acids that have been detected in C. elegans. There have been a lot of papers using GCFRD and GCMS for studying fatty acid metabolism in C. elegans, but we have no idea about the specificity of the individual enzymes in here. Are the LPAs only produced on a very limited set of fatty acid, and the diversity, for example, in the PCs comes from the remodeling in here, or are all of them used? So we don't know anything. We have some example, and this is actually the best studied example that we have. It's MBO7. It's a membrane-bound O-acetyltransferase, which works specifically on the PIs and on the phosphatidylinositose, and it's specifically for the incorporation of polyunsaturated fatty acids into the PIs. So this was a very important paper, but you see it from 2008, and if you look into the data, actually that's a radioactive carbon incorporation into these things, into the different lipid classes. So nowadays with our tools, we can do much better. So this is actually needed in future to really get the specificity that makes also our predictions much better and stops us from over predicting specific lipid species that might be not present or not present at this stage of biosynthesis, for example. One thing we realized also is that we have different fatty acid pools we need for the prediction of these things. So we saw actually that in the C. elegans ceramides, we have typically very long chain N-acyl bound fatty acids in there, typically around C20, 22, 24, and even the odd ones. So C. elegans produce a lot of odd chain fatty acids, so also 23, 25, 27 in there. And they are typically hydroxylated at a second position. So we have there a very different pool from the pool of this of the phospholipids in here. Also there we have no idea how these things are regulated with that enzyme specificity in here. And also again, coming back to the example about this phytosphingolipids, where they are produced. And the last thing, where we have no idea from what's going on at the moment is this N-acyl ethanolamines, and this is something I'm currently very interested in annotating, and I'm looking for data sets where I can find them, is actually these NAPEs. So these are minor lipid species, but they have to be present there. If the hypothesis is true that phosphatidylethanolamine down to N-acyl ethanolamines, that we have this intermediate, and everything speaks for this. So there is in plants, for example, a different synthesis route for such molecules, where it's a direct acylation of the ethanolamine residue with fatty acids. But C. elegans has these enzymes. We have seen this. We have detected this. There are also indications that we have detected these molecules maybe. So I'm still looking for these things. So we have on both sides things that we don't know. It's enzymes producing things, but it's also molecules, intermediates that we haven't detected so far. So we really need to work on both sides. And since lipidomics is getting faster and cheaper now, I hope that in the future we can really go into deep screening of C. elegans, because as a genetically tractable organism, this is a really huge advantage that we have, that we can either get the mutants or do RNAi interference and really check how is this actually interfering with lipid metabolism, and how does this change lipids, species composition, and so on. And with that, I would like to draw a short conclusion. So coming back to my very first slide, worms, fat, and LC-MS actually is fun. So still, after doing this now for 15 years plus, I'm still having a lot of fun, and every new project brings new problems, new issues, and new insights into the lipidome. And as I have written it here, we still have no idea how big the lipidome is, but we're getting better. So from every study that's published, it's getting better and better. There's still plenty of unknown molecules. I didn't talk about signaling molecules like these ascoricides. Frank Schroeder is a big name doing that, but there are also lipid-like structures where we can have up to 30 carbons in the side chains. So still a lot to do. We have no idea about the regulation at all. How is this regulated on a lipid species level? We know some things on class level, but not on the species level. What is the function of these PEGCs, for example, or monomethyl PEGCs? We still don't know. So there are a lot of things, but we have this new data analysis approaches. And now with this really interactive ways of exploring the data, I think having this is getting much faster. For me, I really like that I don't have to shuffle between Excel and R back and forth just to get the plots and all these things and then check the MS. You directly get it in the software for that. One very important thing I like to point out is also user retention times use CCS. I'm a big fan of this orthogonal parameters. A lot of development has been done on the mass spec side, and we're even getting better there. So we are having EAD capabilities in the laboratory, and you might have seen the papers we published with Coral Barbas on the NIST plasma in here. But retention time is really helping you. It's your friend. It's giving you so much valuable information. And one big hope I'm having is that actually C. elegans, it was the first multicellular organism where the genome has been sequenced and as part of the Human Genome Project, alongside Drosophila, E. coli, Saccharomyces cerevisiae, Arabidopsis. So maybe C. elegans can be the first organism where we have also a decoded lipidome. And one thing I didn't cover today is we have it here in the corner with the TIMS HT and the nano LCs. We even went down to single C. elegans lipidomics, so we can now get in a very robust fashion lipidomes of single C. elegans. Because one thing that was fascinating me all the time is they are isogenic, these worms, but it's not like they're all dying at the same day, right? So there is difference. There's are stochastic differences. And the question is this reflected also in the lipidomes? And now we have this tool actually to have a functional readout on a single worm level to get the differences between that. And without further ado, some acknowledgements. So there have been the people, especially Lisa Salza, working on the annotation of the Chipineas, the collaboration with Bruker. Here we stand happily in front of our Bruker TIMS Metabo. And they were always listening to us when we said, okay, you need to improve here on the lipid annotation part. And then the C. elegans community in general. So all the collaboration partners from all the different publications where we work together. Also some people from some annotation hackathons we had in here, for example. So this was a very early one where we're working on a C. elegans metabolic model. And we had people like, you can probably recognize that chap, Michael Witting, which was giving us Lipid 101 on the basics. And this little guy down here, Frank Schroeder, working on, for example, the ascoricides. very, very interactive and a lovely community. C. elegans people are always supporting each other, and I really like that. And just looking to the literature, I recently checked what's going on to curate more lipids. There are even more publications now coming on C. elegans lipidomics. So that's really, really cool for the community. And with that, thank you very much for your attention, and I'm happy to take any questions you might have.
Thank you, Michael. That was great. But before we start, I've just got one question, I guess, before we go to the ones that have been posted. And that was when you mentioned at the beginning about C. elegans, it lives off E. coli. And then I thought, well, when you do the extraction, do lipidomics, I guess you're extracting whole worms. And so,
yes.
And then when you showed some structures, I think I saw some branch chain fatty acids in there as well, which could be microbial. So how do you ensure that you're looking at worm lipids versus microbial and microbial?
So what we're doing is, first of all, when we wash off the worms off the plates, we wash them very vigorously to remove all the adhering bacteria, and also the worm is defecating on them, so it's actually getting rid of all the remaining bacteria in there. But of course, it's a co-metabolism, right? So it takes up the fatty acids in there. And you saw it, right? There were branch chains in there. This is actually one of the best studied things in C. elegans. It can produce actually these branch chain fatty acids on its own. So actually, C15, C17 branch chain fatty acids are produced by C. elegans de novo, and it mostly comes from 99.9% from C. elegans itself for de novo synthesis.
Okay, so it can be, that makes it a real challenge when it comes to trying to work out the biosynthetic origins of some of these lipids. Yeah, very
exactly. Yeah. And we're having huge issues since we don't have a good axenic media at the moment to really culture the worm, because that would be the perfect thing to really study nutrition. At the moment, we always have to have E. coli, and if you want to feed something to the worm, you're always feeding it to E. coli on top, right? Or if you go for other bacteria, you're not have to feed it on E. coli. Actually, E. coli is a very artificial food for C. elegans. It's not the natural food at all in here. But if you would have an axenic media for this, you can take out these things, you can enrich these things. For example, the branch chain fatty acids are coming from the branch chain amino acids. So you can take them out of the diet and see what's happening. That would be really cool, but the worm doesn't like it. So I think we still need a bit of research until we are there.
Yeah, interesting. Okay. So we've got some questions. I'm going to start with Tong Shen. Do you want to go?
Thank you. This is all very intriguing and specialized finding of lipids in C. elegans. So I just want to go back to the biology part. Like, is it certain disorders or treatment we can make it more translational to mammals? Or like other pathways conservative based supported by literature, or we can use it for like environmental toxicology?
Yeah, there are many, many different ways you can use it for, right? So there are a lot of genes where you have homologs in C. elegans. Of course, it's not a human, right? But it's the same true. I could ask the same question. What about the mouse, right? So whatever doing, it's a very simple organism. I'm not saying you can solve all the human problems with C. elegans, but it's a very nice model for basic research, understanding regulation, for example, on a lipid species level, these kind of things. And together with the short generation cycle that you have seen, for example, and it's genetically tractable, you can do a lot of experiments you simply can't do in humans, not at all, right? But also not in mice. So just think about a huge genetic screen. So C. elegans has about 20,000 protein coding genes. So there are huge RNAi libraries, which you can use to screen for specific phenotypes in here. So this is where I see the advantage of C. elegans. So you might have a homolog of a gene for a specific pathway and you want to see genetic interactions. You can do the whole genome screen in C. elegans within a few months, and not if you go for mice, well, that probably takes the lifetime of an entire researcher and the entire group, and even not longer. So it gives you good ideas about things. And then, of course, I totally agree. We cannot directly translate to humans. We have to go through different steps. But it's very good for basic research to get basic understanding of biological principles.
Cool. Thank you.
Brendan, go ahead.
Thank you, Val. Thank you, Michael. Excellent talk. So my question is on the use of the Kendrick-Masti effect, in fact, and it certainly assisted in identifying or annotating many of the lipids. So on one of the last few graphs that you showed, and regarding the trend lines, so there were a lot of points that the trend lines didn't pass through. So those points, those molecules, would they then be still unidentified molecules, or there are other predictions that you have used to identify those data points?
Yes, of course. So this is an explorative data analysis, right? So this is what I want to showcase, these tools that are there. At the end, what you need for good identification would be a standard, right? So since this is sponsored by Avanti, you do a great job in synthesizing things and sending them to us. There could be a few more, especially C. elegans specific lipids in here. But this gives you ideas, right? If you use this Kendrick mass plots, the retention time trend lines, the CCS trend lines, it gives you ideas, right? So what we see, we use nowadays this MassQL, this mass spectrometric query language a lot, where you can formulate specific rule sets. They say, I'm expecting this neutral loss plus this fragments, and if you combine them together, this should be part of this lipid class in here. But sometimes the lipids are just so low that you only get good MSMs for a bunch of them. There might be chimeric spectra because there is something overlapping. So this retention time trend lines actually help us to mine for further members of specific lipid classes, novel lipid classes especially, where we can pinpoint that, okay, we are expecting actually something present here. So we can re-inject things, we can do targeted analysis, targeted collection of MSM spectra for this specific point. So it's a more an explorative thing. It's not that it gives you this definite identification, but it helps you to mine the data much faster.
Oh, thank you, Michael. Thank you.
Thanks, Brendan. There we go. Anne Huang Nguyen, go ahead.
Hi, Michael. Thank you very much for your interesting topic. I have a question regarding the reproducibility in silicon lipidomics. What is your recommendation for the analytical best practice to improve the robustness and reproducibility of lipidomic results? For example, like in terms of sample collection with liquid versus like solid media, or like how we handle the variation when we collect the silicon sample and the normalization strategy.
So the thing is that for most of our experiments, we are collecting an entire plate. So this is normally on a standard agar plate, 9 to 10 centimeters. You can cultivate about 500 adult worms. So first of all, you already get stability by this because you're doing a population average in your lipids. One thing we see actually now being capable of having single C. elegans lipidomics, that things are moving around. So there's much more heterogeneity in the worms that we expected, but this is ruled out on a population average in here. It then depends, and this is getting philosophical about biological replicate, technical replicate. We are doing from the same mother culture of C. elegans different replicates, different agar plates. So the question for the people now is, would you consider this a biological replicate or is this a technical replicate? You could do it on a different day. So what's important for me for reducing the variation is actually, if you're planning an experiment, use the same batch of bactopeptone, agar, and so on and so on. I think this is clear. It's also for cell culture where we are saying, okay, same sample, same batch of things, just to reduce the things. From the analytical point of view, we are using standard Mathias extraction. So I don't think that are a lot of things that could go wrong. I won't say it's a fail-proof protocol, but it's a pretty straightforward one. And one thing I always recommend to my students and postdoc and so on is the KISS principle. And I don't know if you're familiar with it, which says keep it simple and stupid, right? So the more steps you add, the more errors you can do. I can send you our protocols, no problem at all, but they're normally very straightforward. So we use M9 to wash off the C. elegans of the plates. We then do 2 or 3 washing steps, depending also on the type of bacteria we are using for feeding. But this, you remove all the excessive bacteria in here. This is something where we need to actually sacrifice a bit of precision, because normally you say, okay, you want to snap freeze the things immediately. We said, no, we need to do the washing steps exactly for the reason that Val asked, to not have too much bacterial background in our samples. And then we snap freeze or directly extract them, as I said, with standard protocols. We typically spin them down, remove as much M9 as possible, and then off we go. Directly methanol, MTBE, water, and we start with it. So there's no black magic, nothing fancy behind it. And the most important thing is keep it simple and keep it consistent with regards to the buffers and everything.
Thank you very much.
Thank you. So I think one last question, Tony Larson.
Yeah, good. Great talk. Interested in your way of using trend lines with retention time. So we typically use C30 columns rather than C18, and I'd expect the trend line to be similar. How robust are they to things like switches and retention time order for things like isomers? And I guess related to this, what if you use a completely different column chemistry like Hillick? Is there any development in that area for trend lines on retention times?
Yeah, so let's talk about the easy one, reverse phase here. Depends what you're talking about switches, right? So we have a completely different project about retention time prediction together with Sebastian Böcker on metabolites, where we also talk about metabolite switches. And the question is in the order. There are local switches of things that are very closely eluding to each other, right? Where you say, okay, just by chance, one becomes the first one and the other one the second one, so they are switching order. But there are things that are further apart, and if they are switching or if there are differences in there, that might be indicative of really different structures. So we don't use the switches too often. What we know is we take very specific rules that SN1, SN2 elution profiles, one eludes before the others. We know that branch chain typically eludes before the straight chain version in here. But normally our chromatographic resolution is not that good to really have this very, very fine-grained details, right? So I won't say I could separate now a C17 straight from a C17 iso if it's in a complex lipid. Maybe with a lyso-PE or lyso-PC, that's possible. But if you have a PC with two acyl chains, might be not possible. So these trend lines are more like a global trend where you can really see it, where we look into the things and then you see something like, okay, it has the fragmentation pattern, but it's way off, right? So what's going on in there? This might be a potentially wrong annotation or something completely different. So we use it as a filtering device. So we're working on the mass spec side first to establish based on some fragmentation patterns we found ourselves or we curated from the literature to find candidates. And then we're checking if these candidates are falling along a trend line. And then we could potentially identify. Talking about Hillick, I think in Hillick it's even a bit worse because in Hillick you have the elution by classes, and then within a class, of course, you have a bit of separation, but it's not like it's spread out like on reverse phase. Typically, for us, for reverse phase, one lipid class spreads out over 5 to 10 minutes, depends on the lipid class, where you can have good separations. Whereas within Hillick, on the Hillick separation of lipids I did, typically everything is cranked into, let's say, 1, 2, or 3 minutes. And there you have this very tiny trend lines in here. So I think it works better with reversed phase. But that said, I'm not saying it's useless in Hillick. It's just shorter and more condensed.
Right. Thank you.
Okay, thank you. Okay, thanks very much, Michael. That was great. And it just remains for me to thank everybody for coming today. And we'll see you next time, I guess a month from now, for the next Lipid Maps webinar. Thanks, Michael. Thank you, everybody.
Thank you, Val. Thank you, everybody.
Bye.
Bye-bye.